We are now looking for the presentation of Ying Zhu. The title is Deep Spatial Proteomics at Cellular and Subcellular Resolution for Human and Plant Biology.
Thank you for introduction, and also thanks for the opportunity to allow me to share virtually from California of America. So today I would like to share some our my project I focused in the past several years to developing the proteomic technology at cellular and subcellular resolution. And because my focus is more on the technology side, but I do a lot of collaboration with mammalian system and plant system to using the new technology for study human plant cells. So most of projects are done in Pacific Northwest National Laboratory. It's a Department of Energy laboratory. So I recently moved to Genentech and focused on pharmaceutical industry. But the majority of my work presented today is done in PNL. So because this is the genomic conference, so I would like to share some point on because majority of the current microgenomics approach focused on transcriptomics. So it's a very well-established approach, very high throughput. You can measure 10,000 cells. And proteomics is kind of new in this area. So the reason we believe proteomics have a lot of value because protein, as we know, protein is the primary function effectors. And there are a lot of work try to using MRI to predict the protein abundance. Usually it's very hard because the correlation is moderate. Because it's controlled by many different biological processes, including generation degradation rate of both protein and RNA, and a lot of post-transcription regulations to affect the translation of RNA into protein and also measurement noise. So for some certain protein, also it's very hard to use the transcript to predict the proteins, such as extracellular matrix, and also a lot of long-life proteins. So that's a different way to measure proteins. Mass spec only something the new way. The majority of the protein measurement now use antibodies or gene fusion. So as we know, if we use antibody gene fusion, you need looking for a good antibodies. Sometimes it's not trivial, very limited availability. And for plant science, it's more challenging because it's very hard to find good plant antibodies. So that's why I believe mass spectrometry is very valuable for some certain applications, especially for plant science. Because mass spectrometry can directly identify quantified protein without any labeling or gene fusion. And also now we already reach a point we can measure 1,000 protein from a single cell with mass spectrometry. Here is the summarized current spatial proteomics technology. As you can see here, majority of spatial proteomics are using antibodies and imaging like immunohistochemistry and immunofluorescence. Now a lot of new technology try to multiplexing using some labeling and or like MOBI is a mass spec approach, a Codex is imaging approach. So now you can see most of the imaging approach have very high resolution at less than 1 micron resolutions, but usually the target is below 100. And if we want to go to over 100, even over 10,000 of the protein measurement, we have to go using the LC-MS proteomics, and that coupled with LCM or LMD, the laser dissection approach, to isolate the cell and then measure with proteomics. The reason the LC-MS can do deeper protein coverage is the kind of technology do not rely on any labeling. Is just we usually what we do, we have 3 major steps. So we extract the protein from tissue or cells. We cleavage the protein into small peptide. So this get very complex peptide mixture, and they use liquid chromatography to separate all these peptide mixtures, and then measure them one by one using mass spectrometry. So peptide actually identified by the fragment of each peptide precursors. So because we can do it very fast now. Now using new mass spectrometer, we can measure more than 10,000 proteins from single run. A couple hours, we can measure more than 10,000 proteins now. So, but for go to the microproteomics, it's more challenging because the majority of the LCM is developed for larger scale, larger amount of proteins. The major challenge including the low sample recovery. You get some if you get a single cell, you cannot get anything into the mass spec because all sample lost during the cell prep. Also, you have limited LCMS sensitivity for most of the lab, so we have to using the new mass spectrometer have or LC have better sensitivities, and also because the low input of the protein from each sample, the coverage is low, throughput is also low. That's my lab have a focus on in the past several years, try to improve the overall workflow from sample prep to mass spectrometry. The major technology we develop is we call it nanoPOTS. It's a nanodroplet processing in one pot for 20 samples. And as you can see here, the major difference of sample is we do not prepare them into a centrifugal tube or microwave plate. We prepare them in the small droplet on the micro slide, as you can see here. So each the dot here is the colored droplet have about 200 nanoliters. So what do? We do all the sample prep inside each droplet. So that's why we have a liquid pipetting system we build in house. And now actually that's a commercial solution for that. So what we do is, if you look at the video, is we do put a single cell or very low amount of protein into each small vials I can see here, and then using the pipetting system to put a nanoliter droplet contain the reagent of the protein reduction, alkylation, and trypsin digestion, and then finally we use some robot to retrieve them for mass spectrometry analysis. The reason the microfluidics or small volume sample can improve proteomic sample is at first, if you look at the protein coverage of a very low number of cells, so if we prepare them in the tube microwave plate at 25 microliter, it's still very small, but it have a lot of problem for the single cells. We only can get less than 100 protein, 200 proteins, but if we using the microfluidics nanoPOTS, we can reduce the surface sample loss because the protein is very sticky, is easy absorb or any container surface it contact with. So you can reduce the sample loss and get high recovery. And also very important is for single cell, the enzyme concentration is very low. You need enzymatic digestion efficiency very high. So that's why small volume is helps because it actually concentrates both the trypsin and the protein in the small droplet. And other benefit that we realize is also because volume is so small, you reduce the contamination for LCMS. It actually increase the robustness. You can run 100 sample no problem. And also use microfluidics. You can do parallel sample prep. Here is the work we done is do the nested nanopod in one chip. We can do over 500, 700 single cells on one chip. So it can get very high throughput sample prep. A very beginning example is a paper published on 2018. So use the single cell proteomics. We try to understand how the hair cell differentiation in the first couple of weeks of development. So we collaborate with Peter Bagispi in Oregon Health Science University. So the main question we want to answer is the hair cell. Hair cell is the in our every mammalian years. It's a very low number of cells, and also it cannot be regenerated. So hair cell is the cell is mechanical transduction. It's the cell responsible to translate the sound into the electrical signal, so we can hear the voice and signals. As you can see here, what we do is we sort the progenitor cell, we call it supporting cell, and also hair cell use flow cytometry, and then measure them use mass spectrometry. We can generate the pseudo trajectory of the from the progenitor cell to the hair cells. And the most important finding is we found this high abundant TMSB48 quickly degrade during the development. So that's actually give us the confidence why single cell proteome is so valuable compared with other single cell technology, because the actin is actually the protein build the hair bundle for the voice and sound transduction. So in the progenitor cell, actually we see that pretty similar actin amount in progenitor and hair cells, but we really don't know how it really develop into the hair cells. And we measure using mass spectrometry. Actually, from single cell, we can actually calculate the concentration of both actin, the protein, and the TMSD4X. So because a lot of biology already show TMSD can bind to actin at the 1:1 ratio, we do see the same thing as the programmed data type. We can see the concentration is most 1:1. There are very few free actins. But after developing the hair cells, you can see the TMSB4 are most down. So you have so much reacting, you can build hair bundles. You can show here is the example we showed. Proteome is really valuable if we really want to look at how biology works in cells. So in terms of plant cell, we also try to demonstrate using the model cells. Here we use the Arabidopsis leaf, and because that's already well-established approach to isolate chloroplasts. That's very challenging for plant cell because plants all have cell walls. So it's not like a human, you can direct culture them or you can direct dissolve them, use enzyme. So the chloroplast is very fragile. So even we can use this approach to generate chloroplast, we cannot use flow cytometry because photometry just give too much shear force to breaking these chloroplasts. That's why we introduced the cellular one. It's actually the technology we are using a lot for single cell proteomics to isolate chloroplasts. So the very gentle approach can allow us to sort single chloroplast at a very high success rate. So also we can image each cell during the sorting. We know which cell go to which well, and we have image of each. It's very important for us to do troubleshooting. So here is the data we get from single chloroplasts. So here is data as you did peptide, over 10,000 peptide from 10 cells, 3 cells, and a little bit close to 10,000 for single chloroplast from leaf cells. Internal protein, we get 2,000 protein from 10 chloroplasts and 1,800 protein from single one. So it's very good sensitivity for plant cells. And also, as you can see here, is also a lot of studies shows very different from transcriptome. The protein in single cell is very stable. The CV of all the 100 proteins, the CV is at the single chloroplast level is only 0.4%. It means most of the protein show very similar of protein abundance in the single cells. So we apply this approach to study the to understand how the plant responds to environmental stress. Usually we do here in the previous lab. We use the drought stress. It's very easy way. You just water less. As you can see here, we still use the Arabidopsis the plant. So we have a control is 100% water content. We have the automatic system to add the water in the plant. So we have the low drought, and it's 50% for like 2 weeks, and also moderate is kind of much less, 40%. And then we isolate the leaf single cells and then measure the protein change in the single cells. You can see here we do not see much change if you look at color leaf heart, but you can see here for the control low, drought low, it's very similar, very similar profiling from the PCA plot, and even we change it from 50% low water to 40% low water, and the water rate actually you can see the big protein changes start to happen. After we do function analysis on how the liver cells start to respond to the drought stress, so upregulated protein related to the first one is very clear respond to water deprivation, is clear show how the plant responds to the drought stress. Also, see the increase the protein related to the photosynthesis and generate precursor metabolites and also other catalytic activities. It also decrease a lot of protein activities. You can see here is um, a lot of protein related to the ribosome and also structural activity, mRNA binding and translation. It means the plant start to decrease the protein synthesis and also dividing to conserve energy for the environmental stress. So come back to the spatial. So to couple with because all my project is on the single cell, but to do spatial we have to direct work with the tissue. For tissue is we couple it with laser dissection. So here is the system by size. So here is the video. You can see slowly to show how we select the feature and the laser will cut through it, and then we collect directly to the nanopod device we have. So here is the chip, and also we can direct mount the adapters. So we can cut a multi-single cell at the 20 micro resolution and as large as we can 200 micro resolutions. So we have applied to many different applications. At the beginning, it's using brain tissue as a demonstration. It like most of the technology are using, and also we apply to the plant cells. Here is actually metal tissues. We just look at the different cell epidemias, also the inner cell layers. We also can apply the technology to human is human islet, mouse islet, and also uterine tissue. I will give some example here. So for the islet feature, we are looking at it. As we know, islet is a very important organ for all the humans because it secretes all the hormone to control the sugar level. So islet is very small, but it still contains many different cell types. Usually, the major cell population is alpha cell and beta cell. The green one is glucagon is alpha cell, and the red one is serine beta cell. It's well known, but other study of our collaborator really interested is actually is this vasculatures. As you can see here is the FIDC labeled one. It's endothelial cell. It's a cell composed on the vasculatures. Because vascular, if we a lot of study like pharma industry, they develop some drug. A lot of this drug actually destroy these vasculatures, cause a lot of problem of the hormone secretion. So that's the project I want to understand was if we can see the vascular protein difference in the tissue. So we still do the similar things. We section the tissue at like 10 micron thickness, and then we use laser dissection to isolate different cell types. Here we can now demonstrate we look at very different cell populations. The whole islet isolated outside of the islet. And also we focus on two different microvasculature. One is the inner vasculature, is the one we show in previous slide inside the islet. And also we also look at vascular on the surrounding islet. It's the microvascular surrounding the islet. Is interface vascular, like you can see here. And the data we show here is mainly focused on this interesting the same cell type, but because their position is very different. But we see what's the difference of these vascular cells. As you can see here, we see increase the protein abundance in microvascular on the interface in the inner vasculatures. This protein related to a lot of neuron vascular crosstalk. As you can see, this increased abundance related to a lot of neuron transport, neuron snaps, or neuron migration, or neuron specific proteins. That's not this study actually that get a lot of support from public paper recently is how the study try to understand how the human body control the release of hormone actually uses the exome, because the neuron is not inside of the eyelid. The neuron actually is outside the eyelid, but it project into the eyelid using the axon or neural fibers, as you can see here. And neural fiber actually project into the eyelid through the microvasculature. So they're actually together. That's why we see both of them together. So that's actually give a very good example. So we can see validate a lot of previous study using imaging approach. So we also can do something different because we can do dissection. Here is an example to show we can do protein mapping. It's very similar to imaging, but not at that resolution. But we can get a very deep coverage, as you can see here. So we get what we do here different is we isolate each pixel of the eyelid and surrounding tissues. As you can see from 1 to 36, and then we isolate into a single slide and single nanovirus, and then dissect them and digest them into for proteome measurement. Here is the identical protein from each pixel. We can get over 3,500 protein from the 50 micro resolution, and it's very good coverage at this resolution. It's because we only have like 10 cells in each pixels, and we can use this information to understand a lot of something we can see in the imaging approach. So for quick visualize, what we can do is you get the tissue imaging. Also, you can pull out all the well-known protein marker. You can see the insulin enriched in the eyelid area, glucagon is them, and also you can see the low bound MPTX1, and also you can see some ethylene marker is related to a lot of enzyme for digestion, and also we can do some QC, look at the GIPDH. Usually we're using for a lot of quality control for protein asset. You can see they do not have, for GIPDH you don't have the big difference. Of course, we can also use other transcriptomic approach to study deep understand what's the tissue function, but I will not go deep today. So another example is we also can use the technology to look at the cell type specific proteomics for plant tissues. Here, inside of previous PRL, we developed this workflow to collect the we start from poplar, so embedding them into the bending tissue and then section them, and then use the laser dissection to isolate different cell types. So here is the example. We look at the also popular leaves. We look at like the leaf palisade. Here we don't need a lot of tissue because our scientific is much higher. We just look at like one cut and then measure them. So also we can look at the vasculatures, all different cell type. Also for root, we also can do some similar. We can look at epithelial cells, epidermal cell, and also stellar tissue, and also root cortex. So here is a quick example, quick data to show how many protein we get from vasculature from leaf. We get much more, like about 2,500 protein from one cut here. And for palisade, we can get less, but still pretty good coverage at the 1,500 proteins. So for the root, we can get something very similar, pretty coverage. So it's all technology also can be applied to the plant science to understand what the protein different and how the different cell type in the plant responds to the environmental stress that help us to develop a better plant or better agricultural plant. So here is just quickly to do some function enrichment analysis to see what's the difference of protein enriched in different cell types. As you see, for roots, for the leaf, and that's all photosynthesized red protein in the palisade cells. For the vascular cells, there's more carbohydrate and sugar synthesized. And for the root, we see there's more lot of phenolic compound and sugar by synthesized protein in the cortex, and for the vasculatures, we see something similar, a lot of sugar by synthesized amino acid synthesized. That's something is specific in root vasculatures. So this all talk about is about the go to single cell and subcellular level. That's a quick study, quick to show what we are working on now is try to improve the section. What we are doing is we build the deep UV ablation. The motivation to build the UV ablation is one is the current LCM have very limited resolution. It's very hard to get inside the single cells. Usually we can cut the pixels over like 20 micron if it gets smaller, it's harder. And also for plant science, because that's a cell wall, it's very hard to use in the commercial system.
Sorry,
The cell wall.
should
So
conclude your presentation.
the ablation. That's I'm also done. Just last slide. So it quickly show you we can get about 2 micro resolution using the 20x objective and also small pinhole, and you can see even for dye pancreas and we can get 2 micro resolution, and for proteomics we can get over 300 protein from 7 micron. It's a subcellular. It's smaller than single cell. So you can get over 1,000 proteins. So to summarize, I would like to thank my previous team, PNL, and also my postdoc working together to get so many things done. And also the project supported by environmental molecular science laboratory and also a lot of AI project. So I'm done. So I'm looking for your questions.
Thank you very much. If there are questions, otherwise I thank Dr. Zhu and we can pass to the next and last presentation.
Excellent. Thank you.